Okay. Is everyone here? Let me call your name. Check. So we got 23 in total, right?
Yes.
Okay, that's good. So we have everyone here. So we can start the class now. We'll begin with Surah Al-Fatiha. Okay, Assalamualaikum everyone. Thank you for attending the online class. So today this is the week 2. So I'm going to continue from Dr. Rizwan Nur Hakim on the anatomy and also physiology subject. So in this topic, we will cover on tissues and membrane anatomy. So my name is Tunku Faris. You can call me Faris, okay? So for today, we will cover a bit on how the tissues and also the membrane anatomy aspect. Part of it is the eukaryotic cellular organization and functions. Okay, you will learn about the types of tissues that are available in our body, how the cell divides. Okay, we can classify them into a few category. Okay, the first one, you have the somatic cell division. You have also the germline cell division. Okay, the mitosis and also the meiosis. You will learn about the control of cell destiny, a bit on reproductive cell division, cellular diversity, and also the aging and also the cells. So talking about the cells, what did you know about the cells? So we scientists already mapped out what kind of cells available in our body. Okay, part of them is stem cells. Okay, stem cells is considered as the earliest cells in our body. So it has the pluripotent attributes or characteristics. So normally people used to say it's a pluripotent stem cell. Pluripotence means they are able to divide into any kind of tissue, any cell types. Okay, even you can generate multiple cell types from stem cells. Okay, there are 100 trillions of cells in our body. Okay, with thousands of cell types. You have muscle cells. Okay, you have blood cells, brain cells, we call neurons. We have heart cells, cardiac cells. Okay, we have liver cells. What are the other names for liver cells? Anyone? Liver or hair? Hair? Pato?
Hepatocyte.
Yes, okay. Hepatocytes, you have intestinal cells. Okay, there are lots of cell types in our body. Okay, and we also can classify them based on the origin. Okay, the somatic cells, which is the organ cells, and the sex cells, which is the gametes. Okay, we have the stem cell here. It's embryonic stem cells and human-induced pluripotent stem cells. And there are lots of other stem cells. They are still reside in our body. Okay, we have the dental pulp in our teeth. Okay, we have the dental stem cells. We have kidney cells. We have kidney stem cells. Okay, the stem cells are still growing every day. Okay, still proliferating, self-renew, and they are making different types of cell types. And you also need to know that the cells have the cytoplasm, which is the space or the material between the plasma membrane and the nucleus. So two basic elements that you need to know in the cells, which is the nucleus and also the cytoplasm. Okay, the cytoplasm consists of cytosol and organelles. Okay, the cytosol contained lipids, okay, necessary elements for biochemical reactions. Okay, you have salts, you have sugars. So they differs from extracellular fluids due to high concentrations of ions, proteins, carbohydrates, and lipids. Okay, there are lots of essential units and also essential subunits in the cytoplasm. Okay, and you have also the organelles, which is specific structures in the cells that carried out specific physiological activity. Okay, now first we go through to the organelles. This is the basic cellular architecture of the cells. Okay, so this is a cell. Okay, a cell. You have the cytoplasm, which is in brown, and you have the nucleus. And inside the nucleus, you can see here this big rounded dot here. It's nucleolus. Okay, inside the nucleus, which is the location of the chromosomes. Okay, so you have 46 chromosomes, okay, or 23 pairs of chromosome. It's all reside here. Okay, it's all located here. And inside the cytoplasm, this is where you can find all different types of organelles, okay, that carry out the physiological or biochemical activity. For example, you have the backbone of the cytoplasm, for example, the microtubules. Okay, so you need to the like filamentous structure here. You got actin filaments in yellow. And then you have mitochondria. What else do you have? You have centrosome, you have ribosomes, all these things. You have the one this over here. This is the Golgi apparatus. Okay, they are doing an integrative interplay between each other, so they need to communicate uh, from one to another to give a specific signals. Okay, so you begin with the nucleus, and all the information proteins go down to the cytoplasm and being encapsulated inside Golgi apparatus before it being secreted out from the cells. Okay, so the final outcome of this reaction, okay, it could be producing some proteins, for example, hormones, okay, that need to be secreted out from the cells. On the right hand side, you can see that the different kingdoms, okay, have the similar basic cellular architecture. As for example, in the animal, they have a bigger nucleus, okay, but maybe they are lacking some of the microtubules, actin filaments, okay. If you look at the protist, which is the more basic version of the cell, okay, they might lacking some organelle in the plants, okay, very simple, and going down to the simplest version of cell, which is bacteria, archaea, and also yeast. Okay, this is all are the prokaryotes. So we are now focusing on the eukaryotic cells architecture, which is yourself. Okay, we need to know that inside the nucleus, this is where the formation of protein happen. We call it protein synthesis. Okay, it begins with the unfolding of the double helix structure, which is the deoxyribonucleic acid, or simply DNA. So this double helix strand in the nucleus will be transcribed into mRNA, okay, single-stranded mRNA, messenger RNA. Okay, now they are being unfolded, okay, into single-stranded mRNA and further translated, okay, into polypeptide chain or simple amino acid, okay. And this is the outcome of the end of the process. What are these process? From transcription, translation to the protein synthesis. Anyone? We call this as the universal principle of living cell, or known as the central dogma. Okay, at the end of the structure, you will have could be simple version of protein. Okay, you know that protein can be classified into 4 different groups. Okay, it could be the primary proteins, secondary proteins, tertiary proteins, and also quaternary proteins. Okay, if you look at these basic subunits of macromolecular structures, okay, on the left hand side is the nucleic acid. You begin with the DNA. Okay, this is the zoom in, okay, at 1.5 million times. So now it's 10 nanometer scale. You can see the double helix. Okay, but if you zoom out, you can start to see that the DNA actually integrated with proteins, okay, forming a complex. So DNA could not stand alone, so they need some proteins, for example, transcription factors, to bind to the DNA, okay, so that they can start to transcribe information that they receive into the mRNA, okay. So this is the complex of DNA and proteins at the molecular scale. But if you zoom out further, you can start to see the chromatin fiber. All these double-stranded DNA are being surrounding the histone complex, okay, another protein complex, okay. So you can see it's like what's the terminology to describe this? It's being entangled outside, okay, outside the histone complex. So this is the double-stranded DNA. And if you further zoom out, you can start to see the single chromosomes. Okay, so the chromosomes compose of chromatin fibers, which are composed of complex of DNA and protein. And further back down, you can start to see the single subunit of the cells. Okay, it's a very long process to generate a protein up to the stage that they can form the organelles. Okay, all other process or synthesis of organelle follow the same mechanism. Okay, so in the chromosome, you can start to see that there are 46 interspersed inside the nucleus at the cellular scale. Same goes to the protein. If you look at the quaternary structure of the protein, if you zoom out, you can start to see a few blobs of globular proteins, okay, 2, 3 with different colors, and these globular protein arranged in in a good manner, okay, like this one, they forms a layer with the actin filaments and forming the microtubule. So this organelle serves as the backbone of the individual cell subunits, okay. And if you look at the lipid as well, okay, this is the lipid that forms the lipid bilayer with proteins at the cytoplasm membrane. At the cytoplasmic membrane, you can see there are different types of lipids that are available on the surface of the cells. Okay, there are thousands of small molecules or organelles that functions hand in hand just to make sure that they elicit proper signals or stimulus to the next or neighboring cells. Okay, so this is the molecular feedback loops. If you can see that this is the cell cycles, basically what the cell did is to make sure that in every stage of the cell cycles, the cells are ready before push forward and divide. Okay, it started with the G1 phase. Okay, this is the growth in mass. The cells, the DNA start to uh, check for, so at this stage the cells check for DNA needs. Okay, they need to make sure that all favorable environment conditions are um, in the good environment before they proceed to the S phase, which is where the centrosome duplication starts. Okay, if everything goes well, okay, they will continue with the G2 phase. Okay, you can see here now the DNA, two double-stranded DNA will being held together. Okay, and then they will start or proceed with the mitosis. You can see here there's a blue line in the middle that indicate that the cells are ready to divide, and the process of the cell division, we call it cytokinesis. If you look closely to the plasma membrane, as I told you earlier, there are different types of lipids or protein structure, ligands, okay, at the cell surface, just to make sure that the entry or the efflux of chemicals into the cells or outside the cells being controlled, control or regulated properly. Okay, owing to the hydrophobic interior of its lipid bilayer, the plasma membrane is impermeable to ions and most water-soluble molecules. Okay, if you can imagine, if you want to get into your house, you need to pass through to a door. So in order to pass through to the door, if the door is locked, you need to have a special key. Okay, you need to have the key to get into the house. Same goes here. If you need if any chemicals need to go inside the cells, they need to pass through a specific channels, pump, or um, carriers. Okay, right. Same goes with the current situation. If you can imagine how the SARS-CoV-2 affecting or infecting the cells need to have a special receptor. So we call it ACE2 receptor. And the virus cells, start to invade the cells. Okay, they will go to the cytoplasm and releasing their RNA content. So there are various kinds of virus. So generally we can divide the virus into two types: the RNA virus and also the DNA virus. So if it's a DNA virus, the cells or the virus need to um, transcribe into RNA first. Okay, from DNA they need to transcribe into RNA, and it will take some time to give an effect. But in the case of SARS-CoV-2, they have the RNA virus already. Okay, so they bypass the DNA stage. They doesn't need the transcription process because they already have the RNA and they can simultaneously generate a new clones or copies of the RNAs. Okay, that's why they are very quick in terms of the etiology or the pathogenesis. Okay, and once inside the cell, it will start to go to the Golgi apparatus and bind its viral protein there. Okay, and start to kill the cells. Okay, but they will survive and start to infect the other cells. So this is the host cells that will just remain as a factory for them to replicate. Okay, so that they can infect other cells. Have a look on the nucleus. Okay, the most important organelle. You can see the nucleus inside here, nucleolus. You can see some of the chromatin in black. It's not clear because it's electron micrograph. We don't have any color images. I mean real color images for the nucleus. However, you can see there are some pores here at the outside, okay, of the nuclear envelope. Okay, this is where anything could comes in and goes out from the nucleus. Okay, they have like a hole there. Okay, we call it as a nuclear pore. Ribosomes and protein synthesis. So proteins being made up inside the ribosomes. Okay, there are free ribosomes. Okay, floating in the cytoplasm, but most of them attach to the rough endoplasmic reticulum. So you have two types of endoplasmic reticulum, the thin and also the rough. So why its name is rough? It's due to the presence of ribosomes on the endoplasmic reticulum's surface. Okay, this is the rough endoplasmic reticulum. You can see some of the black dots, free ribosomes here, the one they are not attached, but here you can see that some of them attached to the lumen, okay, to the outside of the endoplasmic reticulum. Golgi apparatus. This is the like quite funny structures. Okay, they have the cis phase and trans surface. Okay, when there's a incoming transport of vesicle, they will take it and process it into this Golgi apparatus. Okay, most of the time it's the sugar chain. Okay, they process the sugar, and after that they will being secreted out from the Golgi apparatus. Okay, on the downstream side of the Golgi apparatus down here, okay, processed protein segregate into different vesicles. Okay, lots of vesicles being produced from the Golgi apparatus. Okay, destined for lysosome or the plasma membrane. Okay, they can bind to the plasma membrane, or they can remain in the capsule or vesicle like this as a lysosome. So this containing a processed protein for any kinds of functions. Okay, it could be growth hormone inside here, it could be some insulin. Okay, lysosomes. Okay, it's an impermeable membrane that separates degradative enzymes inside lysosome from other cellular components. If you look at the mechanism, it could be from the Golgi apparatus, it will be secreted out from the cells, or in other way, they could also be endocytosed inside the cells, okay, and from the lysosome, other way around. So that's the mechanism, the two basic mechanism of lysosomes activity. Mitochondria, okay, the fuel, okay, of the or the engine of the cells. You can see that mitochondria enzyme use most of energy released from the breakdown of nutrients to synthesize ATP. Okay, the cell in our body use ATP as the source of energy. Okay, They use this energy for the oxidation of fats, proteins, polysaccharides, okay, into carbon dioxide and water. Okay, so if you want to know that the mitochondria might reside more on the cardiac muscle, okay, or on the skeletal muscles, if you work hard, okay, you can build your muscles. That is where the mitochondria mostly reside. Okay, the area in your body that use most of energy will contain higher percentage of mitochondria. Paroxysms, this is another organelle that you need to know. Peroxy means hydrogen peroxide. Okay, what's the function of paroxysms? It functions to engulf the hydrogen peroxide. We know that hydrogen peroxide is dangerous to your body. Okay, it's very harmful to your body. It's toxic. So the paroxysm will detect this hydrogen peroxide, take it, and then they will turn or convert this peroxide into water and also oxygen. Okay, this is basic mechanism. It happens automatically. You don't need to decide anything. Okay, whenever the cells being exposed to scavengers, okay, to oxidants, okay, so you need to have this paroxysm in your body. Okay, if you don't have some people, okay, they are not fortunate enough to have paroxysm in their cells. So one of the disease, okay, or the genetic defects could be related or causing the mental retardation. Okay, so because the hydrogen peroxide killing the cells. Okay, cytoskeletons and motility apparatus. As for the cytoskeleton, you have the actin filaments, you got the intermediate filaments, some microtubules, okay? They are all around the cytoplasmic space in your cells, okay? They will have the specific functions to hold the organelle in place so they don't move that much. But in other condition, they also functions in motility. You have the cell that contains cilia or flagella. Okay, they have a filaments to sense a signal or to move. Okay, to move something. As for example here, we use it in muscle contraction and sperm motility. And we go to cellular division, how the cell divides. I've told you earlier that the cells at some point, at some stage, they need to be divided. Okay, they need to divide in order to proliferate or to give any specific signals. Okay, it starts with G0 phase, G0, G1, S phase, G2, and mitosis. So this is the phases of the cell cycle. Okay, the interphase, which include the G1 phase, S phase, and G2 phase. The M phase is the mitosis phase, and then cytokinesis happen, and they will continue on and on every day. Okay, replacing a new cells. Okay, replacing old cells, making new cells. As for the cell division, this is mitosis. Okay, the difference is that mitosis happen in the somatic cells. For example, you have the cardiac cells in your heart. Okay, you have brain neuron cells, you have kidney cells, you have hepatocytes, you have skin cells. So all of these bodily tissue use mitosis as their mode of cellular division. Okay, so they divide and generate two daughter cells. Okay, they are totally a duplicate of these cells. Okay, one in red, one in blue, one in red and one in blue. So their total chromosome remain in 46. Okay, Whereas for the meiosis, so for the meiosis, it's undergo meiosis 1 and meiosis 2, but they are not a direct duplicate of each cells. Okay, they have the individual or unique chromatids. Okay, they are not similar to the parent one. Okay, you can see that the parent one have one red and one blue, but here they have only one blue and one red because this is an this segregation happened due to the chiasma. Okay, chiasma here. Can you see that? Okay, this only happened in the meiosis process. Okay, the meiosis process only happened in the germline. Okay, in the gametes or in the reproductive or in the reproduction. Okay, reproductive cells. As in the male is in the testis, and in the female it occurs in the ovary. Okay, so this is the sex chromosome. So this is the process of the meiosis. If you want to know a bit more details, okay, this is the differences. The definition of mitosis is the asexual reproduction in which cell divides into two, generating a absolute replica. Okay, there are no differences with the parent cells. Okay, this is only for the somatic tissue. Okay, as for the meiosis, it refers to the sexual reproduction in which number of chromosomes are reduced by half. Okay, generating two haploid cells. Okay, in mitosis you have 4 stages, while in meiosis you have 8 stages. Okay, the purpose is cellular proliferation, but in meiosis the purpose is to reproduce. Okay, the location occur in all the somatic cells. Okay, heart cells. Okay, hepatocytes. Okay, but in meiosis is located in the reproductive organs. The genetic composition is identical, but in the meiosis it's unique. Okay, the single chromatids is unique. They are not an absolute replicate of their parents. Okay, number of daughter cells produced, 2 diploid cells. Diploid cells mean 2n. Okay, diploid is 2n. Haploid means only n, okay, only one. You have gone through this in your matriculation center, so I guess this is kind of revision for you, just a bit more detail. And the apoptosis. So what are the apoptosis? Okay, apoptosis refer to the suicide mechanism. Okay, when they signal dangerous stimulus, they will start to shrink and burst. Okay, or they will start to shrink and fragmented. Okay, they need to die. Okay, these occur automatically, whereas the necrosis is more due to the external factors or external signals. Okay, if you can see in this picture, okay, when the cells receive the signals that they need to die, they will start to detach from the whole cells, okay, shrink, and then they will fragment it into few more black dots there, and they will lose their function. Okay, and after that, new cells will come and regenerate it. Aging and cellular senescence is different. It's due to the time limits. Okay, as we grow older, people say you become wiser. Okay, but in the case of the cells, we call it cellular senescence. The cellular senescence is defined by irreversible arrest in cell proliferation where cells experience DNA damage at the telomeres and decrease in mitogenic signaling. Okay, you've learned that the cell need to divide, okay, for you to grow. But as you grow older, the cells also grow older, but they didn't lose these properties of replacing new cells. Okay, they retain the properties of making new cells, but the signal they receive okay, the cells still have the capability to divide, but the mitosis signal might be weakened. Okay, so that's the cellular senescence refers to. In tissues, you have to know that the tissues are grouped in different groups. Okay, there are 4 primary types of tissue. The first is epithelial tissue, connective, muscle tissue, and nervous tissues. Okay, they all carry out different physiological functions, and if in fact their structure, their anatomical and architecture are also different, and you can find them in different locations in our body. For example, body coverings, body linings, and in glandular tissues. It functions, so this is the basic functions of the tissues. It provides you with the external protections as well as the internal protections. It functions in absorptions of chemical. It filter any microorganism that tries to invade your cells, and it can also secrete some hormones, for example. So this is the basic functions of the tissue. If we go down a bit deeper at the epithelium tissue, they fit closely to each other, and usually they will form a layer of sheets. Okay, the apical surface, which is the top surface of the tissue, and the lower one we call it basement membrane. It's avascular, refers to no blood supply. Okay, they don't have the blood vessels that much. Okay, and they regenerate easily if it's get well nourished. Okay, this is the structure of the epithelium. For example, here in the top surface, this is the simple epithelium, and they have the basal surface as well. So this is the nucleus. If you look at the stratified version of the epithelium, it forms a few layers here, 1, 2, 3, 4, and 5. And this fifth layer is called the apical surface, and also they have the basement membrane. Okay, squamous cells. Okay, we classify the cells based on their morphology. Some of the cells are cuboidal, columnar. Okay, columnar a bit taller, a bit longer. Cuboidal, a bit like square in shape. Squamous a bit thinner as compared to the other two. Okay, you have the squamous, and inside the squamous you have flattened like fish scales. It's very thin cells. A cuboidal cube, okay, columnar, column-like. Okay, they all undergo specific physiological functions, okay, based on their types. Okay, as you can see here, if it's one layer, we call it simple epithelial. If it's more than one layer, we call it stratified epithelium. And we can further divide them into based on their cell shape. Okay, squamous, cuboidal, and columnar. Basically, they have special functions. Okay, basically absorption, secretion, diffusions of chemicals in and out of the cells. But in the case of more thicker, okay, you have the stratified with the multiple layers of epithelium. They provide the protection. So you have the stratified epithelium, 5 layers, 10 layers. So it becomes thicker now. So its function now differ from the simplest version. Is that all right? Okay. And you need to know that there are also some special cells, we call it transitional epithelium, that functions, that have functions, we call it, I mean, maybe bifunctions. They have 2 functions, okay? For example, it functions in stretching or distending, okay, of urinary structure. For example, in our urinary bladder, when the bladder is full of water, full of urine, okay, it will start to expand, okay, and the cell morphology also expanded a bit. Okay, so these kind of cells, okay, the cell type, we classified it under transitional. So they can change their morphology from the elongated maybe into the simpler one. Okay, I hope that makes sense. Okay, if we go down to the simple epithelium, usually it forms the membrane. Okay, for example, in the air sacs of the lungs, in your lungs, it forms the wall of capillary, and it forms serous membrane that line and cover organs in ventral activity. Okay, you have your lungs here. This is the simplest version of simple epithelia. You have the apical surface, you have the basement membrane too, and you have nucleus in between. And they are very thin. Okay, when it's very thin, it's easier for the penetration of the chemical inside and out. Okay, so this is aspect of lungs. Okay, this is the squamous cell epithelium. You have the nucleus, you have the apical surface, and also the basement membrane. If you look at the simple cuboidal, it's a bit cube-like structure. Okay, cuboidal, cube. Okay, it looks like a cube. So it's usually commonly can be found in the glands and their ducts, in the kidney tubules, and the surface of ovaries. Okay, for example, here this is the kidney cells. You can see that they are simple cuboidal epithelial, so they are like a cube structure. They have the nucleus, like here they have the apical, and also they have the distinct basement membrane. And sometimes in between, you can find the connective tissue, okay, in between, yeah. Okay, it functions in secretion and absorption, and some of them have cilia. Okay, just like the ovary. Okay, they have the cilia to propel the mucus or any oocytes. This is the longer version, so it's a bit elongated. This is the columnar, okay, single layer of tall cells. You can see here, it's a bit taller in shape, and usually it can be found in the digestive tract, okay, some in the mucous membrane, and that line the body cavities opening to the exterior. It also functions to propel the mucus, okay, or any substance, okay, to the exterior parts of our body. Okay, but here you cannot see clearly the cilia, okay, but they are there, right? And the other version is a pseudo-stratified columnar. Okay, the pseudo-stratified columnar also can be found in the uh, what is this? What's the structures? What structures this? Anyone? Anyone? What is this? In blue, can anyone tell me what's in blue?
Trachea.
Trachea. Is it trachea? Is it not esophagus? What do you think? So it says here for respiratory tract. Let me
Esophagus.
check the answer. Are you sure it's esophagus? Esophagus, right? Is it? Because this is for the respiratory tract. Okay. So it has the ciliated version of it, but in a columnar structure. Okay. It looks like columnar, but it's not actually. Okay. It's kind of like fake pseudo stratified version. Okay, let's look at the stratified epithelia. We have the stratified squamous now. Okay, it presents in the skin or outer portion. Okay, mouth and also esophagus. You can see here the stratified squamous epithelium. It's squamous. Okay, but with lots of multiple layers, right? Okay, so it can be found in the mouth, esophagus, and also the skin. You can see that the structure or the morphology of the cells, like maybe 10 to 20 layers, okay, they reside on top of each other, okay, and help in providing the covering or the protection for the friction happen in our body. Okay, it's multiple layer now. Stratified cuboidal, 2 layers of cuboidal cells, functions in protection as well. You have the stratified columnar, surface cells are columnar in shape. There are various size and shape. Basically, it functions in the protection. Okay, you have the stratified cuboidal and columnar version. Okay, you can find both cuboidal and columnar in one tissue, but this is rarely happen, and mostly found mainly in ducts of large endocrine glands. Okay, as I told you earlier, the transitional epithelium composed of modified stratified squamous epithelium. The shape of cells depend upon the amount of stretching, okay, functions in stretching and the ability to return to normal shape. And the only location now found in urinary system organs. Okay. On the other hand, you can see the glandular epithelium as well. There are the cells that functions to form the glands or for the secretion purposes. Okay, it contains proteins. Okay, could be some hormones, some growth factors in an aqueous fluids. Okay, because secretion is an active process, so it will produce the fluid and then make it again and again. As I told you earlier, once the cell receive the information to synthesis whatever protein they have, they start to they start to export it out from the cells, okay, to the extracellular matrix. So basically, you can define, we can classify or categorize gland types into endocrine and exocrine. Okay, endocrine usually ductless. The secretion diffuse into blood cells. All secretions are hormones. Example, for example, the thyroid hormones, adrenal hormones, and pituitary hormones. Whereas for the exocrine glands, they emptied the secretion fluid through the ducts to the epithelial surface. For example, you have the sweat and sebaceous gland, you have liver, pancreas, okay? that functions to produce the insulin. Okay, the beta cell of the pancreas functions to produce the insulin. Okay, and it will include both internal and also the external glands. This is the differences between exocrine and endocrine glands. So if you can see on the left hand side here, this is the exocrine gland. Okay, they exported out the liquid out from the cells, whereas for the endocrines, okay, it will remain inside the body, so it will be secreted into our blood vessel. Okay, for example, the insulin, it will be exported or being transferred into our bloodstream, and then try to reduce the level of glucose in our body. Okay, connective tissue, we can find it everywhere in our body. Okay, it functions to provide protection as well, can bind body tissues together and also supports the body. Characteristic of connective tissue, there are variation in blood supply. Some tissue are well nourished with blood supply. Okay, they're being invaded with lots of veins and arteries, but some cells are poorly supplied. Okay, some of them doesn't have blood vessels at all, so we consider this as a avascular cells. Extracellular matrix, they are non-living material that's surrounding the living cells. Ground substance is mostly water. Okay, apart from apart from lipids, okay, protein or sugar molecules. You have the fibers as well in the extracellular matrix. For example, the collagen fibers, elastic fibers, and reticular fibers. Okay, this is the extracellular matrix environment. This is outside the cells. As for the connective tissue types, you can define it, or you can classify it from based on the rigidity, okay, from the softest to the thickest one, or from the most fluid to the rigid one. Okay, for example, here is bone, which is the hardest material or the hardest organs. Cartilage, a bit softer. You have dense connective tissue, softer, loose connective tissue, and also the blood. Okay, the blood is also considered as the connective tissue. We begin with the bone. If you can see the bone, it has a special morphology. It has lacunae, which is in here. Okay, bone cells in the lacunae, just like the wood. Okay, if you cut the wood into two, I mean the big one, if you cut into two, you can see the similar morphology. With the central canal, it has the lacunae and also the lamella. Okay, it's hard due to the presence of calcium salts. Okay, the calcium salts that make up the bone. Okay, and also presents large number of collagen fibers. It functions to hold your body straight, okay, to support anything heavy, okay, and for you to carry out everyday or daily chores. For the cartilage, it's a bit softer, but uniquely it's situated or located at special place. For example, in the joints, okay, and they are more flexible. In the knee, for example, okay, there are presence of chondrocytes, which is the cartilage cells. As for the hyaline, it's a bit softer than cartilage, okay, but considered as the most widespread type of cartilage. composed of abundant collagen fibers and rubbery matrix. It's all about the percentage of cell types inside there. Okay, it's, for example, if there are lots of collagen fibers, or maybe 80% and 10% bone and 10% connective tissue, so it will define what are the functions that they will carry out. Okay, you can find the hyaline cartilage in the larynx. Okay, entire fetal skeleton and epiphyseal plates, and it functions a bit more flexible skeletal as compared to the bone. Okay, you can see the chondrocyte here. Okay, this is the schematic diagram. On the right hand side, this is the real staining of the cells. You have elastic cartilage. As the name implies, you can see that it functions to provide elasticity. It supports the external ear. Okay, if you touch your ears, it can move up and down whatever direction you wish, and it's provide the elasticity, like in front of your nose, for example. Okay, this is the elastic cartilage. You have also the fibrocartilage, which is highly compressible. It forms like cushion, like a disc-like structure between vertebrae of the spinal column. Okay, the one in purple is the fibrocartilage. Okay, let's move on to the dense connective tissue or dense fibrous tissue. The main matrix is the collagen fibers. You need to know that the fibroblasts are cells that make the fibers. It located in the tendons, ligaments, and dermis. Okay, it's a bit softer, but it's dense. You can see it's a wavy structure here. It's highly packed, but in a wave pattern. Okay, it functions for what are the functions? For the elasticity as well. Okay, for the ligament and also the tendon. Let's move on to the loose connective tissue. Mostly can be found in the areolar tissue. Okay, this is the most widely distributed. It's soft. It's like tissue, like cobwebs. Functions as a universal packing tissue or glue to hold the organs in place. Okay, it's also layer of areolar tissue called the lamina propria that underlies all the membranes. Okay, the bottom here in red, we call it lamina propria. And all these fiber types form a loose network, and they are easily detached from each other. Okay, they can be cut easily, but they provide the elasticity for the movement and gluing everything so that everything should be in place. Okay, it can soak up excess fluid that causes the edema. Okay, it can hold the water. Okay, before the water can go up to the other tissues, but then it needs to be held at the lamina propria location. As for adipose tissue, its metric is an areolar tissue in which fat globules predominate. Okay, many cells contain large lipid droplets with nucleus to one side, call it signet ring cells. Okay, you can see here the fat cells. The fat cells is very fat. Okay, to the point that it pushes the nucleus to the periphery. Okay, these fat cells contain the lipid droplets. So this is the schematic diagram. If you look at the right hand side, which is the real image, pathological image, you can see they appear in white that contain the fat droplets. It pushes the nucleus to the periphery. Okay, it functions to insulate the body in terms of cold or different temperature. It protects some of the organs as well. Okay. Reticular connective tissue, mostly in the spleen, you have a delicate network of interwoven fibers with reticular cells. The morphology look like the fibroblasts, but they are not. The locations in the internal organs, such as the lymphoid organs, spleen, lymph nodes, and bone marrows. Okay, so this is the lymph vessel. Okay, the lymphatic vessel where you can find some of the white blood cells. Okay, the functions to produce most of the uh, lymphocytes. Okay, for the immunoprotection. As for the blood, we consider it as the vascular tissue. Okay, the blood surrounded by fluid matrix known as the blood plasma. Soluble fibers are visible only during clotting and functions as the transport vehicle for cardiovascular system, carrying nutrient waste and respiratory gases. Okay, blood is also classified under connective tissues. You can see the red blood cells or erythrocyte here in capillary, neutrophils, the white blood cells. Okay, it's bigger as compared to the white blood cells that don't have any nucleus. The monocytes, okay, that can engulf any pathogens. Now you have been taught about different cells, organelles, okay, cytoplasmic components and tissues. What are their functions? So this is just to summarize that inside the tissue, there are the combinations of organs, there are combinations of cells that functions to protect or to filter or to secrete any kind of chemicals, okay, or maybe as a defense mechanism for our body. Okay, here you can see that there are reticular fibers, as I told you earlier. They support in blood vessel walls, forming branching networks around the various cells that include fat cells, smooth muscle cells, and neurons. What else? You have some plasma cells that develop from B lymphocyte to secrete antibody and for attacking the antigens. Okay, you have fat cells here, you have the blood vessels, okay, you have collagen fibers. So they works synchronously to protect or to maintain our body homeostasis. Look at the muscle tissue. It functions to contract or shorten or to produce movements. You can see that there are three different types of muscles: skeletal, cardiac, and smooth muscle cells. As for the skeletal, it is voluntary. Okay, it's up to you if you want to use your muscle. Okay, you can decide where to use it or where not to use it. And it produces broad gross body movement or facial expression if you do anything related to the skeletal. Okay, characteristic, it has the stripes, lots of stripes, multinucleated, more than one nucleus, long and cylindrical shape. Okay, can you see the cylindrical shape? The cylindrical is horizontal, not the vertical one. Okay, it's very tiny there. Okay, cylindrical shape. As for the cardiac muscles, you have the muscles on the cardiac cells, on the heart. Sorry, okay, it's involved involuntary movement, so you cannot control it. You cannot control the rhythm of your heart. Okay, it pumps the blood through the vessel, and it also can be recognized by its features. The shape contains stripes, but they have special or unique only one nucleus. Okay, you can count it. It's not like the skeletal muscle where you can find lots of nucleus. Okay, the cardiac muscle is a bit different, uninucleated, short and branching cells. You have the intercalated disc containing gap junction to connect cells together. And finally, you have the smooth muscle cells. It's also involuntary, you cannot control it. It's found in walls of hollow organs, for example, the stomach, uterus, and blood vessels. It carried out the movement of the vessel, okay, through the mechanism we call it as peristalsis. And characteristic of smooth muscles include uh, invisible striations. You cannot see any stripes like cardiac muscles or the skeletal muscles, right? It's one single nucleus and it's spindle-shaped cells. Okay, it looks like spindle. Okay, brain cells, or other words, neurons. Okay, it functions to receive and conduct electrochemical impulses from and to our body parts. Okay, it functions during irritability and conductivity. I don't know what these two means. Okay, um, maybe just keep this for now. I don't know what does the irritability means here. What does things that make it irritated? Okay, but you just need to know that the nervous tissue will become excited after after they reach a certain um, electrochemical impulses. Okay, it also contained some of the accessory cells. We call it support cells, called neuroglia. Okay, that protect and supports the neuron. So this is like the accessory, but you still need to have it in the nervous tissue, or else they cannot provide the good environment for the transfer of the signals. Okay, for example, so this is the picture of the brain, spinal cord, and the nervous tissue that contains the neurons and also the supporting cells. Okay, the neuroglia. Okay, so in this class we have covered some basic introduction to the different types of tissue. There's functions for the internal communication, for example the brain, spinal cord, and also the nerves. You have the muscle tissue for the movement and supports that have the bones, hearts, and also smooth muscle for the hollow organs. You have the epithelial tissue that protects your body linings and also provide um, the location for absorption. Okay, provide the medium for the absorption. You have the connective tissue that supports, protects, and bind the tissues together. Okay, so this is basically your whole system that comprise all these essential tissues that conduct a specific systems. As for the regeneration of tissues, in the case of cuts or injury, the tissues also have a special mechanism for them to recover or regenerate the cells from the injury. So as for example, if you can see here the epithelial tissue like skin and mucous membrane, okay, they are highly regenerated. Okay, they are very active. They can respond very quickly to any injury, and some of them are regenerate poorly. For example, the skeletal muscles, if it's injured, okay, it will take more time for them to recover back to the normal functions, and some of them are cut and replaced with a scar, so they will leave the scar tissue. For example, the cardiac muscles, nervous tissue in the brains, and also the spinal cords. Okay, they will have an effect on the anatomical structure of functions, and the growth through the cell division will continue, okay, through the puberty. Cell populations that expose to frictions, okay, also the epithelium will replace the lost cells throughout the life. Okay, you have the connective tissue that remains mitotically active every day. Okay, it will repair the cells without you ever knowing it. Okay, but with some exceptions, the muscle tissue will become amitotic by the end of the puberty. Okay, some of the muscular tissue or muscle tissue, they can no longer divide themselves at the end of the puberty. They have lost their properties, so it will depend on the remaining cells to carry out the functions. Okay, as for the nervous tissue, it becomes amitotic shortly after birth. Okay, amitotics refer to the inability to conduct a cellular division. Okay, nervous tissue can no longer actively divide after delivery. In the tissue repair for the wound healing, okay, whether the regeneration or the scar occurs depend on the type of tissue. Okay, if it's a important tissue, so it will take longer time. Okay, or it will leave the scars. Okay, in the case of epithelium or the cells that are regenerate easily, it will take lesser time. Okay, clean cuts, incision heal more successfully than rake tear of the tissue. Okay, okay. If you cut yourself with a knife and compare you yourself the X. Okay, the X for example. Events in tissue repair. Okay, it started with inflammation. Okay, where the capillaries become more permeable, and then the clotting proteins start to migrate towards the injury site, and then they will form a clot at the injured area. Okay, the next process involves the growth of new capillary. Okay, phagocytic cells dispose of blood clots and fibroblasts, and they can rebuild the collagen fibers. So it starts to granulate on the tissues. And also in the next step, which is the regeneration of surface epithelium, this is where the scalp detaches, and whether the scar is visible or invisible, it depends on the extent of the wound, okay, the severity of the wound. The more you get injured, the longer time for them to regenerate um, uh, and healed. As for the tissue development, there are terminologies that you need to know. Neoplasms can be classified into two groups, benign and cancer, or malignant. Other words, yeah, for cancer, malignant, it represents abnormal cells, masses that cells cannot control cell division. Okay, in terms of benign, the cell uncontrollably proliferate but still under control. But in the case of malignant or cancerous, they are highly active and proliferative to the extent that they can no longer be in control. So they are over proliferation of cell division. Okay, and this cancerous cell can metastasize or invade the neighboring tissue or the bloodstreams. Okay, that's why in the cancer you can find some of them, even though they have received the chemotherapy, but still have the remaining cells that are not responding enough to the therapy. Okay, even though they have being considered healed, but the cells are still infected with the cancerous cells. In hyperplasia, as the name implies, it refers to the increase in the size of tissue or organs. Okay, it may occur when the tissue is strongly stimulated or irritated. Okay, the cell number become increased. Atrophy, decrease in cell size. Okay, tissues or organs when the organ is no longer stimulated normally. I would like you to find the example of diseases to each of these condition. Okay, if you can come out with the example of cells that uh, undergo hyperplasia, okay, what are type of diseases, and also the atrophy, where you can find this atrophy. Normally, where does this condition or pathological condition occurs? Okay, let me know. Neoplasm, hyperplasia, atrophy. I guess this is for your own readings. Okay, you need to know all these three examples in the real life. As for the Islamic view, we need to know that all these things, all the biological, chemical, environmental, physical, physiological, everything in our body is being um, arranged or managed carefully by the architect, okay, which is Allah Subhanahu wa Ta'ala. We know that the cells could not produce proteins on their own. Okay, it's Allah who determined how and where they're gonna be synthesized. Okay, of course, we have the will to decide what we're gonna do, but our will is depend on Allah Subhanahu wa Ta'ala. Okay, the cells can mutate, become a disease cells. Okay, um, if Allah doesn't allow it to happen, okay, it won't happen. Okay, Allah is the one who created all these cells in a fashioned manner. Okay, the cell is the only place on earth where the entire range of life sustaining biochemical reactions can function. So an unbroken lineage stretches from the earliest cells to each living organism. Okay, if Allah wants to disturb one of your nucleotide base in your DNA, you can get the mutation. All right, but how Allah can manage in our genome that the DNA can transcribe into RNA according to their time, and from the RNA to the specified protein accordingly. Okay, this is Allah's creation, so we need to appreciate that. I think that's all for today. It's quite a lengthy presentation, but I hope you have the basic ideas. What are the functions of the tissues, the functions of the organelles, where they are located, and you need to know why Allah created this specific tissue at that location. Okay, I think that's all for today. We will continue next week with some other lecturers. I'm not sure who they're gonna be. Um, but this is from my part. My advice is, if you need the soft copy of the presentation, kindly email me at faris@gmail.com. Sorry, faris double s @iium.edu.my. Okay, because I don't have your email address. So if you can kindly email me for the lecture notes, then I can send it to you. So this is my email. Okay. Any questions from the audience? If you need to ask anything, if you're not clear. Okay, is anything question? Okay, there's no issues. Okay, I hope all of you will stay healthy at home. Okay, do study and read more books every day if you want to know more about the anatomy and also physiology subjects. Okay, kindly let us know if you need any help. Okay, whether you are not having good internet connection or anything, we were more than happy to help. I think that's all for me now. Is there any question? No. All right. I think that's all. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Doctor.
Thank
Thank you,
you.
Thank you,
Doctor.
Doctor.
Thank you, Doctor.
Thank you, Doctor.
Doctor.